Bismillahirrahmanirrahim. May we start another paper, 9700 As Biology June 21, paper 2-1. This is going to be the next three questions, the question 4, 5 and 6, and this is the second video on this paper. Now starting with question number 4, we've done the first three questions in the first video. Now this is the second video. Question 4a says the induced fit hypothesis and the lock and key hypothesis are used to describe the mode of action of enzymes. Explain the induced fit hypothesis. Now the induced fit hypothesis is what? It is that the active site is not fully complementary to the substrate. The active site changes shape, or you can say molds around to fit the substrate. Or you can say, of course, these are conformational changes for the shape change. And this results in the enzyme substrate complex being formed, and the active site then returns to the original shape and releases the product. And change of shape, in fact, results in lowering the activation energy and puts strain on the bonds. And so it is the catalytic site or the active site where all this is happening. So the lock and key hypothesis is the opposite of this. That is, there is no change in shape, but it actually complementary to the substrate and it fits into it. And of course, the R groups on the active site and the substrate interact, and that results in the catalysis taking place. So the enzyme works in that way, induced fit and lock and key hypothesis. As you can see here in the mark scheme, active site not fully complementary to substrate. Active site changes shape or molds around to fit the substrate. And there's a allow, it says A means allow conformational change for shape change. Then number 3, enzyme substrate complex form. Now you see here ESC is allowed, enzyme substrate complex forms. So this is an abbreviation which is allowed. Then active site returns to original shape on release of product. And then any valid point, number 5 is any valid point, change of shape to give complementary fit lowers the activation energy or puts strain on the bonds. And so the binding site and the catalytic site of the active site. So please understand how you read these mark schemes. It's very important how you read these. Now there are 5 points, but any 3 would have got you the 3 out of 3. So it's not that you have to write all these 5 points to get the 3 marks. You have to give any 3. So if you could have written maybe the first point and the third point and the fourth point, well, you would get your 3 marks. Now coming to the B part of the question, radish plants contain the enzyme peroxidase. So there is one enzyme mentioned, peroxidase. So we would, if you said enzyme, we would know you're saying a peroxidase. That catalyzes the breakdown of hydrogen peroxide. You know hydrogen peroxide is what? H2O2. Students investigated the effect of increasing the concentration of hydrogen peroxide. So hydrogen peroxide is a substrate. So we have concentration of hydrogen peroxide in millimoles dm cube. Look at the units. Always look at the units. Please remember that. So the concentration of hydrogen peroxide, so this is actually the substrate on the activity of peroxidase extracted from radish. The results of their investigations are shown in figure 4.1. Initial rate of reaction in micromoles per minute. So we see an increase and then we see it becoming constant. The initial rate increases, but then the initial rate after this point becomes constant, and it is at these concentrations which you will read off here from the graph, in which it has become constant, but it is increasing, but then the increase is, there's a sharp increase or then there's a gradual increase. Now part 1 of the question, explain the effect. Explain is biology, it's not describe. Explain the effect of increasing the concentration of hydrogen peroxide on the initial rate of reaction as shown in figure 4.1. So I drew a very simple example. I say this is the enzyme, say this is the enzyme peroxidase, and you have 100 of these active sites. And then of course you have the substrate, which is in this case is of course, here we had this enzyme was peroxidase, here there was hydrogen peroxide. So say you have 10 of these. When the reaction starts to take place, well, only 10 enzyme substrate complexes were formed. When you increase it to 100, then of course more enzyme substrate complexes formed, more product formed per unit time. But when you increase this and you increase it more or you increase it more, then the active sites become the limiting factor. You have so much of the substrate, but then there are no active sites available for the reaction to take place. So enzyme becomes the limiting factor. But in this situation, when this was the story, when you had only 10 of these, then the substrate is the limiting factor. When you have 100, now of course, all the active sites are occupied. And now even if you increase it, the active sites are not increased. So the rate becomes constant. The rate becomes constant. Now let's look at the mark scheme, 4B1. Peroxidase, it says A means allow peroxidase for enzymes. So if you said enzyme, we knew you were talking about peroxidase. So any three, rate of reaction increases as substrate concentration increases 0.3 to 0.35. So up to 3.3 to 3.5 mole millimole dm cube, some active sites are not occupied. So that's why the rate is increasing. Some active sites are not occupied. More collisions between enzymes and substrate molecules is taking place. So more collisions means more enzyme substrate complexes formed. Now this is for the part in which the rate of reaction was increasing as the substrate's concentration was increasing. Now as it levels out, here we have another situation. Rate of reaction remains constant between this area. Now this is the part which we are explaining, this part. So all active sites occupied, saturated. A limiting factor here. At low substrate concentration, when we were talking of this part of the graph, then the substrate was the limiting factor. In this part of the graph, the enzyme is the limiting factor. And then the limiting factor, when it plateaus at high substrate concentration, enzyme concentration becomes limited. Substrate is not limiting anymore. So out of all these 5 points, you had to give me any 3 to get your 3 out of 3. Coming to question part number 2, the students determined the Km for radish peroxidase as 0.10 millimoles dm cube. With reference to figure 4.1, describe how they determined the Km. And that's 2 marks. Always look at the marks when you're doing the question. So if you look at the Km, how did you figure this out? What you have to understand is you've got to calculate the Vmax. Once you've calculated the Vmax, then you have to calculate what? You have to calculate half the Vmax. So when you do half the Vmax, so if you've calculated half the Vmax, so then you calculate, then from the half the Vmax, you're going to look at, say half the Vmax was 3. It's not 3, actually, it's something, it's 5.5, so 2.7, so somewhere here. And then you're going to read off the graph. So the Km is the concentration of the substrate concentration at half Vmax. Is the substrate concentration at half Vmax. So it has told you that it is 0.10. But they said, how would you do it? So how you do it? There's only 2 mark scheme points. First, you determine the Vmax. So you determine the Vmax, which is 5.6 micromole per minute. Right? So you determine the Vmax, and then Km is the substrate concentration. Km is the substrate concentration at half Vmax. Okay, that's it. So everybody knows what is Vmax, everybody knows what is Km. Then it says a further investigation found that the Km for carbonic anhydrase is 12 millimoles per dm cube. Describe the role of carbonic anhydrase in the transport of carbon dioxide. So it is nothing to do with the Km, but they're saying describe the role of carbon dioxide. And this is 3 marks. So the role of carbon dioxide. Now let's look at that. So if you look at this question, the role of carbonic anhydrase in the transport of carbon dioxide, you're going to be talking about 2 places, one in the respiring tissues and one in the lungs. Now in the respiring tissues, carbonic anhydrase catalyzes the reaction between water and carbon dioxide in red blood cells. It forms carbonic acid, which dissociates to form HCO3 negative and H positive ions. HCO3 negative moves or diffuses through membrane proteins from the red blood cells into the plasma. Then it maintains a concentration gradient for diffusion of carbon dioxide into red blood cells. Because if the carbon dioxide remains in the red blood cell, the diffusion gradient will finish. So carbon dioxide keeps on diffusing in and is being converted into carbonic acid, so it's always low inside the red blood cell. This helps to remove large quantities of carbon dioxide. Now what happens to it in the lungs? In the lungs, the carbonic anhydrase converts the HCO3 back into carbon dioxide and can be exhaled out. So the reverse reaction is catalyzed. So carbon dioxide can be excreted out. Now if you look at the mark scheme of 4C, in respiring tissues and in lungs, so there are 2 different places where you're going to be talking about. In respiring tissues, the carbonic anhydrase catalyzes the reaction between water and carbon dioxide in red blood cells to form carbonic acid, which dissociates into HCO3 negative and H positive ions. HCO3 negative diffuses into the plasma through the membrane proteins from the red blood cells, then maintains the steep concentration gradient for diffusion of carbon dioxide into red blood cells, then helps to remove large quantities of carbon dioxide. Then in the lungs, the carbon anhydrase converts the HCO3 negative back into carbon dioxide, so can be excreted or removed or exhaled. So these were the 6 mark scheme points, and you have to give me any 3 of these. Question 5. Much success was made in reducing the number of cases of malaria between 2000 and 2015. Explain how malaria is transmitted. How is it transferred from one person to another? If I have malaria, how can I give it to another person? Vector is the female Anopheles mosquito. Mosquito takes blood from an infected person, and then the mosquito inserts the saliva with the pathogen into the blood of an infected person. But actually the detail of it is that when the mosquito sucks the blood of an infected person, then the blood goes into the stomach of the mosquito. Then from the stomach, the pathogens with the plasmodium migrate to the salivary glands of the mosquito, where they multiply. And then when it has to suck the blood of another person who may be infected or uninfected, it can be either case, then it has to inject its saliva, which contains an anticoagulant. So when it injects the saliva, the pathogen is in the saliva. The plasmodium is in the saliva. So by mistake, the plasmodium is also transferred. It's not done on purpose. The mosquito doesn't know that what it's doing. But because the saliva contains the plasmodium, so the plasmodium is transferred to an uninfected person. So please understand that, but you know how to word it as well. Then it says diagnostic test strips for malaria contain monoclonal antibodies. The test shows the test strip detect antigens produced by the pathogens that cause malaria. Figure 5.1 shows the stages in the production of monoclonal antibodies. The information in 3 of these stages is incomplete. Complete figure 5.1. Malarial antigen injected into mouse. Malarial antigen injected into mouse. Immune response occurs in mouse. Then there's a gap. Specific what? Specific what happens? Specific B lymphocytes. So this would be B lymphocytes or B cells. Specific B lymphocytes or B cells then divide to form plasma cells. Plasma cells extracted from spleen of mice. Myeloma cells. Plasma cells dash with myeloma cells. Fuse with myeloma cells. Or you can say combine or join or merge. Please do not use the word hybridize. That's not a correct word. That's not allowed. And what does it result in? It results in the formation of a hybridoma. I've written it in a bigger manner, but of course my marker is different, but of course you will try to fit in your answer in this space. So results in a hybridoma. So this would be the 3 marks. Then the part C of the question. Figure 5.2 shows 2 diagnostic test strips for malaria. A sample of blood from a person suspected of having malaria is put into the well labeled S. So this is the well labeled S. So we put the blood from a person of malaria here. Sample S for sample. A buffer solution is put into the well labeled A. So we put a buffer solution here. The buffer solution moves the blood towards the result window. A line at position C indicates the test is working correctly. A line at position T. So C means it is working correctly. But if there is a line at T, then it means a positive result for malaria. The person has malaria. State 3 advantages of using test strips for malaria such as those shown in figure 5.2, and that's 3 marks. Now if you look at the mark scheme for 5C, results are accurate, rely on monoclonal antibody, results are obtained quickly, diagnosis very quick, you don't have to do a blood test, put the microscope, put the slide, look at the slide, it's going to take a lot of time, you're going to need very professionally trained people. Then is low cost and cheap, does not need highly trained medical professional, does not need specialized equipment, easily transported, distributed in bulk, then can ensure appropriate treatment is given immediately malaria is confirmed. So it will be easy to do, everybody can do it anywhere, everywhere can be done. Reduces unnecessary use of anti-malarial drugs just in case. Only a small sample of blood needed. and can identify different species of Plasmodium. So it was a very quick, you have to think of it, that was a very quick way, it was easy, it was cheap, and all those things didn't require any professional people to do the test. To the D part of the question, the highest number of cases of malaria occur in sub-Saharan Africa and Southeast Asia. Discuss the factors that determine the global distribution of malaria. Now, this is a very syllabus point, the global distribution of malaria. So, now let's discuss this. Mark scheme, they have given you different headings for it. They've said, let's talk about the habitat of Anopheles mosquito. Now, it is usually, mosquitoes breed in warm, tropical, humid climatic regions. In areas where Anopheles mosquito vector occurs, Anopheles mosquito only live in humid conditions. Warm temperatures for the development and growth of the Plasmodium and the mosquito. Warm temperatures for the development and growth of the mosquito larvae. Then mosquito require bodies of still water for breeding. So you need ponds or puddles or lakes or swamps. Then mosquito require places where there is sufficient rainfall because rainfall is going to result in, then after the rains there are going to be puddles of water everywhere. And then of course it's got to be low altitude where the mosquitoes survive. They don't survive at Murray or Switzerland or places where at a higher altitude. Then resistance, insecticide, then resistance of the mosquitoes to insecticide, then drug resistance of the parasite. If a lot of insecticides are being used, then the resistant forms of mosquitoes start to develop. Then prevention in countries where prevention control measures are not implemented by government and health authorities. Then what control measures, like for instance, there is a lot of water standing everywhere, or they don't spray, they use chemicals to kill those, or they don't use insecticides. So if prevention is not doing well, so of course, then of course, that means they are poor countries and they can't afford to spend money on the prevention of malaria. Then immunity, then in the population, immunity to malaria and human population. If the people are immune, then of course it limits the distribution of the disease. Then migration of infected people from area with high rates of malaria. So for instance, we had migration from people from Afghanistan. So if there were certain diseases which they were bringing to us, then our people would also start getting those diseases. So migration of infected people from those areas, from other areas where there is malaria, rampant malaria, and then it goes to other places. So they would be carrying the malarial plasmodium. And then high rates of HIV infection. If you had more people with HIV infection, they are immunocompromised. So they will result in opportunistic infections. So these were the 15 mark scheme points, but you had to give me any 5 of these to get your 5 out of 5. Now coming to the last question, lysosomes are cell structures that contain enzymes known as acid hydrolases. Figure 6.1 shows some processes that occur in animal cells. Now let's look at it. There's a bacterium which they have labeled. So you can see these bacterium, and these are the bacterium which they have labeled. And then you see C, and then you see D. You see the bacteria, then you see the bacteria being taken up, and then you see B something happening, D something happening, and then they've labeled lysosome, and then they have labeled E, and then they have labeled F, and then they've said not to scale, so it's not a very scale, it's not up to the scale, so things have been drawn not in the right scale. Now it says name the cell structures labeled A and E. So what was A? A is the rough endoplasmic reticulum. Please do not write this in abbreviation because sometimes that is not allowed. So rough endoplasmic reticulum. Rough endoplasmic reticulum. Please get the spelling right. I'm sure you all know the spelling. Rough endoplasmic reticulum. Then what was E? Now E, as you see, is a stack of flattened sacs. Golgi body. So Golgi body is not one word. Sorry, I might have written it as if it appears as it's this one word. It's not one word. It's two words. So Golgi and then body. So Golgi body, or you could have said Golgi apparatus, or you could have said Golgi complex. Any of these was correct. Then it says state the function of the structures labeled F. So something is budding off and then it's going to the Golgi. So labeled F transports from the ribosomes to the Golgi. So transport from the ribosome to the Golgi. So this is all what you had to give for 1 mark. What does it do? Function of the structure labeled F. It's a secretory vesicle, and what is it? Is transporting that protein which was made on the RER, and then it is being transported to the Golgi. So this is all what you have to write. Name the process by which bacteria are taken into the cell at C. Well, whatever it is, it's very simple. Phagocytosis. Phagocytosis, or you could have said endocytosis. That's all what you could have said. Then coming to the D part of the question, with reference to the processes occurring at B and at D in figure 6.1, outline the role of acid hydrolases in lysosomes. Acid hydrolases, what do? They break down or destroy the bacteria. They destroy the organelles, or if you're going to say the named organelles, or some organelle which are worn out organelles. And they catalyze or they speed up the hydrolysis. Then any named two substrates, you could have said polysaccharides or proteins or nucleic acids. And then these biological molecules can be recycled or reused within the cell. And then the macrophages and cut up to present the antigen. So any of those three, and you got your 3 marks. So D part was simply giving me the functions of the lysosomes. As you can see here, break down, digest, or destroy the bacteria. These are the mark scheme points. Break down, digest the worn out defective organelles, or you could have said a named organelle, like you could have said mitochondria. Then they speed up hydrolysis, hydrolase. So they speed up hydrolysis. Then any named substance, like you said, proteins or polysaccharides, because bacteria are made of peptidoglycans, so they were breaking that up, polysaccharides or proteins or phospholipids or even DNA, nucleic acids. So any two named substrates. Then the fact that they can recycle or reuse the biological molecules. Like for instance, if the cell membrane is broken up, then the cell membrane is going to result in phosphates and the glycerol and the fatty acids. So these could be reused. If the nucleic acids are broken down, then the nucleotides can be reused by the cell. So reuse the biological molecules within the cell. Then the phagocytes cut up to present the antigen. Yes, the macrophages becomes the antigen presenting cell. So the macrophage or phagocyte cuts up to present the antigen and become an antigen presenting cell. Now coming to the last part of the question, and the last question that ends this paper. Carrier proteins in the membranes of lysosome maintain a lower pH than the surrounding cytoplasm by moving hydrogen ions. Suggest how the carrier protein maintains a lower pH within the lysosome. This is the lysosome and we have a lower pH. Means there are more hydrogen ions. You know, pH is power of hydrogen. So what is it going to do? It's going to move, or it's going to pump the hydrogen ions into the lysosome. So hydrogen ions are going to be pumped into the lysosome. This is the lysosome. So hydrogen ions are going to be pumped into the lysosome against the concentration gradient. And this, of course, is going to be by active transport. And you know active transport requires what? Active transport requires energy. And that would, of course, be using ATP. So energy from respiration or ATP, and this would result in a conformational change of the carrier. There is going to be active transport. So this is the lysosome. Hydrogen ions will be pumped into the lysosome. So more hydrogen ions means a lower pH. So for 2 marks, this is all what you have to give me. Pumps hydrogen ions into the lysosome. Pumps hydrogen ions into the lysosome. Against the concentration gradient. 1 mark. And this is active transport. Got you the 2nd mark. If you said active transport, or if you said uses ATP, there's an alternative mark here. It's not the same point, please remember. Or if you said energy from respiration. You didn't need all of these things. You just needed one of them. If you said active transport, well, there you got the mark for you. Okay, this finishes this question paper, and all I can say is thank you for watching, and best of luck in the coming exams.